So this
Yes.
is hello. Any problem? So guys, please go on mute at this moment. So guys, welcome to this presentation, this seminar. So first of all, let me introduce myself. My name is Priyam Pandit, and I am a MPharm qualified candidate student professional. So I have passed out BPharm in the year 2016 from BCDA College of Pharmacy, and after that I have passed out MPharm in the year 2018 from Jadavpur University in pharmaceutics. So after that I have worked in QC department for 6 to 7 months, and then I joined the pharmacovigilance field. My first company was Cognizant Kolkata. I have worked there for 2.5 years, and after that I moved to IQVIA Bangalore. So currently I'm working as operation specialist 1 or senior associate in IQVIA, and I have a total experience of 3 years plus in pharmacovigilance. And with me there is Shogata, who is sharing the screen. He is a student from Bharat Technology only. He has passed out BPharm in the year 2018, and he is also working in IQVIA with me. So first of all, this seminar, this topic is on pharmacovigilance. So I would like to tell you something about pharmacovigilance. So this pharmacovigilance subject has always been underrated actually, because when I was in BPharm also, we didn't have any subject or any topic on pharmacovigilance in our entire curriculum. So different people used to come and they used to present lot of things, but they have never I have never seen a people or a person who has presented something on pharmacovigilance or clinical research. There are a lot of people coming from QC production house, but there was no one from pharmacovigilance field, and people do not have basic idea about pharmacovigilance only. They think that pharmacovigilance is nothing but a basic data entry only, which everybody can do. But in actual scenario, the thing is not that simple. Yeah, definitely data entry is there, but the thing is not that simple, not simple data entry. There are a lot of other things, lot of other knowledge or areas of expertise which you need to explore for joining a the pharmacovigilance industry. And I don't know, maybe WBUT has introduced this pharmacovigilance course in their 4 years curriculum at this moment. So, and it is very important basically, because what we have seen that when we just joined after interview, means somehow we are cracking the interviews, but after joining the industry, after joining the production environment, it was like a tremendous pressure or huge pressure. The training was not that simple, because we didn't have any idea about pharmacovigilance apart from the definition that clinical trials or those adverse events related things which is present in Cology books. But there are a lot of things associated with pharmacovigilance which we need to just learn after getting to after joining the production environment, after joining a company. And the training time frame is also very short. That means in a very short time they make us learn lot of things, and it becomes very difficult actually. So this knowledge of pharmacovigilance is very important, especially if you want to grow up your career in the field of pharmacovigilance. The pharmacovigilance basically is a desk-based job, and the difference between pharmacovigilance and normal production or QC job is that, since I have worked in both the fields, I am telling you, in production or QC, you need to, there will be a lot of physical effort. Means you need to just run and work. You will not get an opportunity to sit even, especially in QC. For entire day means for 8 to 9 hours you need to keep on running and standing and working. But pharmacovigilance, it is a desk-based job where you can sit at a particular place and you can work peacefully, comfortably, but there will be a lot of mental stress, or you can say that you need to apply your brains. There are a lot of things to apply. So let's move on to the presentation, and I will just show you the journey of this pharmacovigilance. I will start with basic definition of pharmacovigilance, and after that what kind of work we do in pharmacovigilance. We mainly do ICSR case processing, which is the core activity of pharmacovigilance, and today I will just try to show you how this work is done basically. So what are the things which you need to know before cracking an interview? What are the questions you can expect from an interviewer of a pharmacovigilance? And what are the companies which are providing job opportunities in the field of pharmacovigilance? So these all things I will just show you today in this short presentation. And we have a channel called Soulmate Pharma. We are just working on this channel. We have a lot of videos and we provide topics related to pharmacovigilance, provide discussions on pharmacovigilance topics, as well as we also provide job updates in the field of pharmacovigilance on weekly basis. So if you people are interested to get those updates, you can visit our channel. I am pasting the link in the chat box. So this is the link of our channel. So if you want a more elaborate or you want to know more about this pharmacovigilance course, and you want to stay updated with the job opportunities in the field of pharmacovigilance all over India, then you can visit this link. So moving on to the first topic. So Sogata, yes. So this is the introduction part. So we all know the definition of pharmacovigilance. So the term means drug, medicine, and vigilance means watchfulness or awareness. So it is a branch of science that deals with awareness on medicine. So as per WHO PV, pharmacovigilance is the science and the activities relating to the detection, assessment, understanding, and prevention of adverse events or any other drug-related problems. So it is saying that pharmacovigilance deals with detection, assessment, understanding, and prevention of adverse events or any other drug-related problems. So the point is that it is not only detecting or concerned with the adverse effects or adverse event which we experience after taking a product, but it is also concerned with any drug-related problems. Now, what are drug-related problems basically? Drug-related problems are some of the problems which are associated with manufacturing defects. One example is manufacturing defect. For example, you are taking a product, you are taking a tablet from a strip, and you find that the tablet is defective. One of the tablet in the strip is defective. It has orange peel effect or some other mottling or some other sort of defects, or the strip is also, the labeling is incorrect, or the strip is also tampered. If you find such sort of things, then also you can report it to the concerned medical marketing authorization holder, or even you can report to regulatory authorities. So reporting is allowed here. Then medication errors. You must be knowing about administrative error, dispensing error, prescription errors. So these any sort of medication errors which are being, for example, if a prescription is illegible and for that the pharmacist has dispensed a wrong medication and you have received a wrong medication and you have taken that wrong medication, so that you can also report. That will be also be taken care of by the marketing authorization holder. So it is not like that you will be reporting only adverse events only, but you can report anything. For example, if you experience of someone is experience car accident or motor vehicle accident after taking a product of marketing authorization holders of a particular company, and you can report that event. The logic behind reporting is that they will just check whether whether the drug of that particular marketing authorization holder, whether it is anyhow responsible or for causing any sort of problem in that person, in the physiology of the person or not, whether it has caused any problem or not that may have lead to the accident. So they will just investigate. And this product quality complaint and manufacturing defects, these are some things which are not submitted to the regulatory authorities, but the company just they keep a track on this, and they will just rectify their mistakes in future. So for that, they always encourage the patient to report everything which they experience. So even a silly thing you can report, and that will be taken care of by the marketing authorization holders, and they will see, if they will further check whether that has to be submitted to the regulatory authorities or not. They will accordingly evaluate, but your report will be acknowledged. Whatever you are just reporting to the particular marketing authorization holder. So it is not only reporting adverse event, but any sort of drug related problems or any problems which you have experienced after taking a drug. So as per EMA, that means European Medicine Agency, which is the regulatory agencies of European countries, as per EMA guidelines of GVP, preventing harm from adverse reaction in humans arising from the use of authorized medicinal products within or outside the terms of marketing authorization or from occupational exposures, and promoting the safe and effective use of medicinal products, in particular through providing timely information about the safety of the medicinal products to patients, healthcare professionals, and the public. So it is just not based on the products which are available in the market. You can see the definition states that authorized medicinal products within or outside the terms of marketing authorization. So for example, a drug which has not entered phase 4, which has not entered the field of pharmacovigilance. Pharmacovigilance means phase 4. If a drug is in phase 1, phase 2 or phase 3, under these phases, during that time, lot of patients or lot of healthy volunteers are selected for clinical trial purpose. And during clinical trials also, if some sort of adverse experience are experienced by those volunteers, and if those adverse events are also needs to be reported. So regulatory authority also needs those reports, and based upon this, they will just approve whether a drug needs to be marketed or whether it is safe to be introduced in the market or not. So lot of people have a misconception that pharmacovigilance, this field, is only based upon post marketing surveillance or phase 4 studies, but it is not like that. There are a lot of cases, lot of companies, even we work on phase 1, phase 2 and phase 3 clinical trials as well. Reportings are equally welcomed or done for those 3 phases as well. Those things also needs to be reported as well as documented. And the concerns of PV have been widened to herbal, blood products, biological, medical device, vaccines, traditional and complementary medicine. So not only allopathic medicines, but pharmacovigilance is also applicable for herbal medicines, blood products, different sera, antisera, toxoids, biologicals, medical devices, vaccines. So nowadays COVID-19 vaccines were invented, and due to this there was a huge surge in pharmacovigilance industry. So in the past 2 years there were a lot of openings in pharmacovigilance, and the companies have provided a huge salary as well, because lot of projects were coming and they were just concerned with vaccines. So reporting of adverse events associated with vaccines are also important, and those are also submitted to the regulatory authorities. So whenever there is a plague or some sort of epidemic or pandemic, or when there is a sort of new disease and new medicines are being developed, then there will be a surge in pharmacovigilance industry for jobs. So this is the basic principle of pharmacovigilance. So whenever there is a demand, there will be more job. Whenever there is a new disease, whenever companies are shutting down, IT sectors are shutting down, and due to some sort of any sort of disease or any sort of health issues, then people will develop, pharmacists will develop will engage themselves in developing medicine as well as, and then due to this the pharmacovigilance industry will bloom. There will be more job opportunities available. So Shogata, please move on to the next slide. So now I'll say why pharmacovigilance is important basically. So pharmacovigilance is basically required because in simple phase 1, phase 2, phase 3 studies are not enough alone. Those studies in clinical trials cannot provide you all the information which pharmacovigilance will provide. So without pharmacovigilance or phase 4 study, phase 1 to phase 2, phase 3 studies will be incomplete. So tests in animals are insufficiently predictive of human safety. So we know that first of all in preclinical trials before phase 1, animals are used, and animals testing on animal, their physiology, their ADMR profiles are not equivalent or not like that of humans. There are a lot of differences. Though mice has lot of similarities, mice, monkeys have lot of similarities with the humans, but it's not like that they are totally similar. There are a lot of differences between their physiologies. So that's why there is a need of doing human safety studies. And in clinical trials, patients are selected. The population is usually clean. That is, exclusion criteria are limited in number. So for a clinical trial, to conduct a clinical trial, we require a lot of patients actually. So for phase 1 we need 20 to 100 volunteers. For phase 2 we need 100 to 500. For phase 3 we need around some thousands. And so for this, even in these thousands of people, so if an experiment is performed on thousands of people only, so that is not sufficient enough, because whenever a drug is introduced to a market, crores of people will be exposed to that particular drug, and people from different regions will be exposed, from different countries. The geographical variations the geographical variations among the people will vary according to the country, and and also their physiological alterations will also vary. So conducting a trial in just a very small number of people in a very small patient base, or under a people with where the population is around few thousand, it is not sufficient enough to draw a conclusion. So it is very much important to conduct a trial in an expanded patient base. And conditions of use in trials differ from those in clinical practice. Definitely in trials, when we are just conducting a trial, the environmental conditions, the laboratory conditions, sometimes the conditions are just stimulated. These are not natural conditions, not exact environmental conditions which are prevailing. So the in vivo conditions, in vitro conditions are basically different actually. And likewise in conditions which are present in lab or in real life conditions are also different. So that's why it is very much important to do practical experiment or to keep on continuous monitoring when a drug is introduced in a market under normal environment conditions. And durations of trial is also limited, though clinical trial takes years to conduct, or it takes a lot of years, around 12 to 13 years are already wasted. Even more than that are just invested for clinical trials, but still that is not enough, because time changes, the geographical condition, weather changes, and the microbial resistance starts to develop. A particular strain or particular disease starts to develop a various resistance to a particular drug, and as a result this trial needs to be continued. So these pharmacology or pharmacists, they just keep on altering the structure of drug, they just keep on altering, making modifications to the drug molecule, as well as they are just to cope up with the change of which a disease strain is just undergoing. So if a disease strain is undergoing a particular change, then accordingly the drug needs to be altered as well. Their structure activity relationship of that particular drug needs to be altered, needs to be reanalyzed, and if necessary some new modifications needs to be made so as to cope with that particular disease or particular change which a organism is undergoing. And information about rare but serious adverse reactions, chronic toxicity used in a special groups such as children, the elderly or pregnant women, or drug interactions is often not available or limited. See, the biggest problem in clinical trial is that lack of human volunteers. So it is very difficult to just search for the volunteers or who are just ready to participate in clinical trials, especially during higher phases, phase 2 or phase 3, where there needs, say, we need patients for a particular trial to conduct. So it is very difficult to just search for those patients actually, because different people have different problem and they just don't want to participate, because participating in clinical trial is always risky. You may lose your life. So definitely nobody will want to participate in the trial, especially if the study is chronic. That means it is undergoing for a long period of time. So people will not have that much of patience to stay or to just stay in the trial continuously. So they will just try to voluntarily or involuntarily withdraw. They may just escape or run away, and it is not possible for anybody or anyone to just, we cannot imprison that person for just being into the trial. We cannot force that person to be in trial. So that is the basic problem and the biggest problem which we experience for the clinical pharmacist or the person who is taking the trial, sponsors, they just experience while conducting a trial. So lack of volunteer patients or volunteer participants is the biggest problem. Then next slide. So now I will just show you a brief idea about the history of pharmacovigilance. So how this pharmacovigilance started basically. So if you just see the era, the history of pharmacovigilance, it started around 170 years ago in January 29, 1848. In what happened, a girl named Hannah Gira from England died after receiving a chloroform anesthetic. So she was suffering from infected toenail, and a doctor just administered chloroform anesthetic to operate the person. And chloroform is a powerful anesthetic, and the dose of the chloroform was not optimized at that time. He just deliberately gave a certain amount of chloroform, and during that time the pharmacovigilance was not developed. There was no such sort of clinical trials, no such regulations, and even humans, there were no consent before a trial to take place. So they just used to introduce a medicine or any sort of drug to a person randomly without informing that person, or they used to just torture humans or just torture humans or the associate people inhumanly for a clinical trial. So without letting someone know about a trial, it is definitely illegal for nowadays, because laws are strict now. But the journey was not so smooth. There are a lot of sacrifices. Basically, lot of people have just lost their lives due to this clinical trial, due to this lack of informed consent. There were due to this lack of regulations. The first was this infected Ronilwala incident, in which a girl just received a chloroform and she just died. The cause was initially unexplored why the girl died, but after that it was invented. It was found that the death of the person was due to administration of chloroform at higher level, or the dose was not regulated at intoxicated dose, which led to the death of that girl. Next, there was another incident where sulfonamide was used in 1932 for treating streptococcal infection. So a syrup was introduced, and in that prontosil or sulfonamide was used. So sulfonamide was used to treat the infection, streptococcal infection, and a syrup was introduced. In this syrup, diethylene glycol was used as a solvent, and diethylene glycol is a toxic solvent. The taste and odor of the solvent was evaluated, but the clinical safety was not evaluated, and due to this, there are 107 deaths. So after that, this Federal Food, Drug and Cosmetic was established. That means first regulatory agency was established, which came into force in 1938, and its aim was to renovate the public health system. The next was Thalidomide Incident. We know this. It was the biggest move, and this was the very, very, very fatal incident, and which just altered the market or moved the people. So thalidomide was first marketed in 1957 in West Germany, where it was available over the counter. So thalidomide was used. Thalidomide caused phocomelia. It was administered for morning sickness or motion sickness, but it caused phocomelia when it was administered to the pregnant woman. But that was not previously explored. So, and it remained uninvented basically, that thalidomide is causing phocomelia in pregnant women. This was not investigated, or this was not made ever before. So the around total number of infants affected by the use was 10,000, of which 40% of died around the time of birth, and those who survived had limb, eye, urinary tract and heart problems. So, and it was removed from market in 1961. So just see the gap. It was introduced in the market in 1957, and it was removed in the year 1961. So it was almost 4 years, and until 4 years it was introduced in the pregnant woman, and as a result, lot of child lost their lives, and most of them were just taken birth with congenital anomalies. So next slide please. So now I will just show you some of the dark side of human experimentation. So initially I will just say that it's not like that human experimentation was not there. It was there, but it was like the human experimentation was conducted in inhuman way. So it was not mandatory, but it was conducted somewhere, but it was conducted in inhuman way. So it was just like a torture basically. Especially the torture was done with the prisoners. Even the drugs were introduced, the drug under development, these were introduced in patient population or people even under the school child without letting the school child know. Even they were, even the prisoners, the condition of the prisoners were the worst affected. For what happened basically during war times, when the war was going on, people were just imprisoned, and those imprisoned people were just taken out. Their limbs were being amputated, their eyes were being extracted without giving a particular anesthesia. Means live dissection was performed. They were just dissected without any sort of informed consent, and they used to just crave for death basically. Because see, if someone is just taking out of your limb or hand, just cutting with a sword, just cutting it with sword your limb, and you are just alive, then the pain you will just feel basically, your area will, that the cut area will get infected. Even they were not just, the people were not putting any sort of medication to the cut area, the area will be infected, you will just become septic, your sepsis will just spread everywhere, and the person will die. That means the death will be a very painful. So the experimentation was done in that way only. There were no regulations, and people were just being slayed down inhumanly. So from surgeries without anesthesia to clinical trials performed overseas, the history of human experimentation is riddled with stomach-turning movements. So medical testing were done without consents. Jenner injected Phipps with the smallpox virus, where it might have killed the boy. He was unfazed. From this experiment, Jenner created the first smallpox vaccine. So the journey, the smallpox vaccine came to the market in a very, not in a smooth way. Just see, a person just introduced smallpox virus to a particular boy, that particular boy was dead, and the experiment was done in this way. So this was the scenario basically. Inmates of California's San Quentin State Prison were subjected to numerous medicinal procedures, including receiving transplanted testicles from recently executed prisoners. So the testicles of the males were removed, and the transplanted testicles were introduced basically. So this was very painful. Many men received transplanted testicle organ from rams, goats, and boars. Just see, the testicle from animals are being introduced to humans. What a horrible scenario. Children with cognitive and intellectual disabilities held at the Willbrook State School in Staten Island, New York, were given hepatitis or trusted source, so that doctors there could track the spread of the viral infection and how it responded to globulin injections. More than 700 children were infected. So just as I have already said, children with disabilities means the person who is suffering from disabilities, who are physically challenged, those persons were tortured, because the concept was that they are good for nothing. So they were just infected with any sort of infections, any sort of diseases, and experimentations were done. And due to this, many people have sacrificed their life. So next slide please. So now live dissection has been used as a form of medical exploration in Japan, and estimates of thousands of people were subjected to medical, biological, and chemical weapon testings. The prisoners of war, along with Russian and Chinese citizens, were operated on without anesthesia, had limbs amputated to study blood loss, were injected with animal blood, spoon to death, and infected with gonorrhea, syphilis, plague, cholera, smallpox, and other fatal diseases. Even children were not spared. Eyes were injected with chemicals, legs were amputated, surgeries were performed without anesthesia. So actually I have seen lot of short films. I have also seen a Japanese short films where I have seen that in Japan the prisoners are like that. Nowadays the prisons are like that, the poor people of Japan, they deliberately do some small crimes to just get into the prison because they receive a very good treatment in the prison. Their prison in Japan are very aristocratic. They provide lot of facilities and they will provide almost VIP treatment compared to the prison of Indians or any Bangladesh or any nearby countries. But initially, in initial days, the scenario was not like that. There were inhuman tortures in Japan, Russia, Chinese, in China. So in those countries, there were inhuman tortures to the prisoners. So due to these sort of problems, due to these sort of experimentations or clinical trials, such inhuman tortures, there were lot of just objections, lot of revolutions, and ultimately some declarations were passed, laws became strict. Declaration of Helsinki, which is one of the most important declaration concerning to informed consent. So informed consent, you must have heard in clinical trials, where the consent of human, their written signature is very important for conducting a clinical trial or asking him or her for clinical trial. So these all sort of declarations or regulations came into the market, and nowadays the laws are very strict. Regulatory authorities are very strict. Even for a small adverse event or small sort of problem also, if a drug is found to be responsible or if it is found that the drug is causing too much of harm, a company may require to provide a huge compensation, huge fines are being imposed, or ultimately the drug may be banned from the market as well. So next slide please. So now moving to the core PV. So what is pharmacovigilance? What forces we do in pharmacovigilance trials, pharmacovigilance field? So we have discussed about definition of pharmacovigilance, clinical trials, the idea of clinical trials, the history of trials, the painful experimentation. Now we will see what we are just doing in industry basically. So this is basically a technology transfer type of presentation, and we will just focus on what we are doing in the industry, what activities we do basically in pharmacovigilance. So in pharmacovigilance, we just do adverse event study reports. So we just see at what are the adverse events, we just collect those information, and we just submit those reports. We just submit the reports in two ways basically. Some reports are submitted in expedited way. Expedited reporting, as the name suggests, the reports should be submitted within a very short period of time to the regulatory authorities, means within some 15 days. Basically the idea before to submit this expedited report is that because since the drug is already available in the market and regulatory authorities, they don't want to take any delayed decision whether a drug needs to be done with the future, and it is very much important to let people know, or the physicians or the person, let those person know how this drug is performing in the market, because accordingly they will just prescribe those drug, or they will just prescribe the drug to the patient population. And so it is very important to just conduct a study or just to submit the report to the regulatory authority in a very short period of time. So expedited reporting is important. Particularly expedited reporting is done for those drugs, for those reactions which are unexpected. Unexpected means it has not taken place before, and it has not taken place before with that particular drug, after taking that particular drug, and the event is serious. So for those all sort of drug candidates, expedited reporting is that. And there is another sort of reporting, that is periodic reporting. So in periodic reporting, what is that? This is basically aggregate reporting, where after a particular period of time, after a certain amount of time, the reports, the adverse event experiences are being collated, and these are submitted to the regulatory authorities, maybe after 6 months or after 1 year, as per the regulatory authority guidelines of different countries. So these are submitted to different authorities, and this aggregate reporting is done for those adverse events which are not that serious, which are basically non-serious, or which are non-serious in the sense that it is not causing some sort of basic very vigorous or very bad adverse event. It is just causing a silly adverse events, and which is not life-threatening or fatal, or it is not causing any permanent anomaly to the person. So those sort of adverse events are considered for aggregate reporting. And also there are some other adverse event, and if the event is serious but it is expected, that means a particular adverse event, it has already occurred during its clinical trial or after a post-marketing, after being introduced into the market. If a particular adverse event, serious adverse event, has already occurred to a particular patient, and if it is reoccurring to another patient, then that event is known as expected. That means it is already documented somewhere. So we have basically reference safety information, that document where all the adverse events related informations are present. That is known as reference safety information, where all the study observations, what all observations have been observed, what adverse event, what are the warning, contradiction, precautions, what all things are happened to a particular drug after it has been introduced in the market or when it was in clinical trial. All these are documented in a document, particular document, that is known as reference safety informations. So those, if the event which is occurring to a particular patient is already documented in that particular reference safety information, then the event is considered to be expected or labeled or listed. So guys, if you want, you can just sit with a paper and pen and you can just note it down, because these terms are important. Basically there are a lot of new terms which are important for your interview as well, because interviewers, they will just ask the questions from these terms only. So as I was saying, so if the event has already occurred in a particular patient population before with a particular drug, then the event is known as expected. And a particular event which has not occurred before, then the event is considered to be unexpected. So now next, moving to the signal detection. So signal detection is basically nothing. It is the potential adverse event or potential adverse drug reaction which occurs after taking a particular drug to determine. So for example, after taking a drug A, patient is experiencing headache. So this headache is a new signal. So that is, headache is a new signal. That means headache has not occurred before. This is a new adverse event, and in signal detection, those adverse event is analyzed, and we just go for a causal relationship. So causal relationship basically means to detect the temporal relationship, whether the event is basically associated with a particular drug or not. So basically we know the difference between adverse event and adverse drug reaction. Adverse event, so adverse drug reaction means any untoward or reaction which has occurred after taking a drug, but that particular reaction has a causal relationship with that particular drug. That means that particular reaction, it is due to that particular drug only. If it is established, then it is known as adverse drug reaction. And however, if you are experiencing any sort of incident, for example, any sort of accident, car accident, motor vehicle accident, any sort of medication error, or any sort of any physiological alterations in your body, any sort of things which you are experiencing after a particular drug, but that causal relationship, whether the drug is responsible for causing that particular event or not, if it is not clear or it is not clarified, then it is known as adverse event. So this is the basic difference between adverse drug reaction and adverse event. So, and this is an important question for freshers in pharmacovigilance field. You can say this is a patented question which almost every interviewer asks: what is the difference between adverse drug reaction and adverse event? So this definition is very important, and our pharmacovigilance, the journey, one of the important activity of pharmacovigilance is to go through that particular journey, from particular journey of adverse event to adverse drug reaction. We just explore and we just see whether the adverse event is considered, whether the adverse event is particular or it can be considered as an adverse drug reaction or not. So we just do different algorithm, different experimentations, different maths are also there. So different calculations are being done and different factors are considered, and based upon these factors it is finally concluded that no, this adverse event is particularly adverse drug reaction, or this adverse event is not an adverse drug reaction, and accordingly we establish a temporal causal relationship. So this is the basic idea of causality assessment, which is the most important criteria, most important work in pharmacovigilance. And based upon signal detection, based upon this causality assessment, we just go for risk management. So risk management, basically, as the name suggests, to identify what are the risks, what are the potential adverse drug reactions associated with the particular drug, and we just explore the risk benefit ratio. You must have read in pharmacology, risk benefit ratio, the analysis is done. So based upon that analysis, the marketability of or the future of a drug is basically decided. So if the risk of a drug outweighs the benefits, or if it is found that the risk is higher than that benefit, then a drug can be withdrawn from a market. So the company may receive notice from the regulatory authorities to withdraw the drug from the market, or the company may need to change or make modifications to the structure of the drug, to the activity of the drug. They may need to do a structure activity relationship to change that particular drug molecule, so that benefit outweighs the risk, not that risk will outweigh the benefit. So that is the concept of risk management, to explore, to identify the risk benefit ratio and to ensure that whether the benefit is higher compared to risk or not. Now basic steps. So now I will just let you know what we do in pharmacovigilance after joining any company. If you may be in Kolkata or any company, what activity we will need to do. So what you will need to do daily, regularly, because these things are not being discussed basically, and these things are not, people don't know about their job, what they are doing basically. So basically I will just explain this in a very simple layman language, so that it will be easier for you. See, consider a practical example. When there is any sort of government job or any sort of application form, you are just applying for a job, or any sort of interview, and for that you need to, or any sort of online examination, you need to fill up an application form. So for filling an application form, what you do basically, you take certain documents with you, because you need to provide, you just keep some sort of documents in your hand, because lot of information will be asked when a computer, you will just open or click on the link to fill up the application form. The screen will appear, lot of tabs will be there, where you need to fill your details, your parents' details, then your qualification, your experience, your photo, and etc. Lot of things needs to be filled out. And for filling those information, you will require some sort of documents with you. You will just see those information or collect those information in document, or sometimes it is better if you just write all those information in a particular paper. In particular paper you are writing those information, and you are just seeing that paper and just filling up the information. Or you may just take all those information, all the documents with you, and you can just start fill those necessary information, your details from those document. So you will fill those information. You will just, after filling all the necessary information, what you do, you would just click on a page, or finally a page will appear where all the informations will be assimilated, or a collated information will be present in a particular page, where your name, details, etc. will come in a single page, and there at the bottom a declaration is present. I confirm that this information is correct to the best of my knowledge, and no changes can be made after this once we submit the application. So in this way declaration comes, and we just see that particular page, that draft page, and verify all the informations whether those informations are correct. What we have filled tab wise, those information are correct or not. Once those information we tally, once those information are being verified, we just click on submit. We just save and submit those information, and after that the application form, or the particular application form, is submitted, and we take the printout of that particular form which we have verified, or the particular application form, and we keep it with us as a document evidence. So the activity in pharmacovigilance is somewhat like that only. So here the particular document which you will be using to fill all those information tab wise, those particular documents, that particular document is known as ICSR form, or individual case safety report form. That means this form will contain all the information related to a particular patient. So the patient will just provide their name, details, address, phone, then their demographics, then what sort of diseases they have experienced, what is their lab conditions, means what lab test he has undergone, what product he has taken along with the suspect product, or the company suspect product, then what adverse event he has experienced. So all this information he will just provide in a particular form. Using that particular form, we need to database, we need to just, with Pharmacovigilance Association needs to just database those information in various database available. So we just need to fill up those information. So that particular form is known as ICSR form, or individual case safety report form. This definition is also very important for interview purpose. And once those information is filled up, we just generate another form which is known as SIOMS form. I was saying that we just see a form where we just find all the filled up information, whatever you have filled up tab wise, we just filled up, we just see all those information collated in a particular form, that is known as SIOMS form. We just need to see, by checking the SIOMS form, we just need to ensure whether the informations which we have filled up is correct or not, because tab wise checking is definitely very difficult. So that's why a collated sheet is provided, a draft sheet, where we just check whether the information is correct or not. And once we find that the information is correct, or if required, if we find that any mistake is done, we can edit those things. We can go to we can visit that particular tab, and we can just make those changes. And once those changes are done, we just again just see that particular SIOMS form, that form, and we just submit that report. The submit option is there. Here the submit is not, here in pharmacovigilance we just don't consider it as submit, we just use the term release. We release that case. So once the case is released, That means now it will go to the regulatory authority for submission. That means now we cannot make any changes. That means it is full and final locked. And with us, we have that particular COMS form available. So that it is equivalent to that printout application form which you have submitted. So this is the basic idea of pharmacovigilance. This activity we basically do in pharmacovigilance. Now you may think that this is very simple, very easy. Nothing is there just to see, just to fill up all those information by looking at the document and just to submit this. But the scenario is not like that. It is not like filling only those information, but you need to apply lot of medical knowledge, lot of medical sciences. You need to explore lot of things before just filling those information. Because see what happens, basically patient, they are not, means I mean to say that patient, they will not have that much of medical knowledge, so they will report things in a very, means they will just report it in a very unscientific way or unprofessional way. So they may use very common terms or layman languages. So sometimes they just report in this way, that for example, I am just saying that one example, simple example. They may say that I was in such a problem that it seemed that someone was hammering on my head. The pain was such a severe that it seemed that someone was hammering on my head. They use this sort of English languages. So what happens, basically, hammering on my head, pain was the severe. So it is, we need to just analyze what is the medical concept behind that. We cannot submit in this way to the regulatory authorities. We need to explore, we need to just apply our medical knowledge, because regulatory authority needs a very strict and very professional reports. They will not entertain any sort of this sort of unprofessional or this sort of layman language wala reports. So as a pharmacist or as a healthcare professional, we just need to convert those layman language wala information, and we just need to impart or input or identify the basic medical knowledge which is associated with that particular event. So that is the basic idea. So our job is to that identify that particular concept, medical concept. So I will just explain these things in some subsequent slides, or you can just refer to our YouTube channel link as well to get a more idea on these things, because this is a very huge topic. Lot of things are there and we cannot cover up all these things in a short period of time, basically. So basically we just use two databases. Again, the name of the databases are also important. We use ARISG tabs as well as Argus. These two databases are widely used in the pharmacovigilance field, in the pharmacovigilance industry. Although there are a lot of databases, in fact there are a lot of customized databases. Companies, they can also develop their own database, but two databases like ARISG and Argus are basically used in the market. So ARISG consists of the following tabs. I have just pointed out the name of those tabs. What are those tabs, what are present? So first contains general, then patient tab, then product tab, then event tab, then narrative tab, causality tab, labeling, that lab, study. Whereas Argus also consists of these tabs only, but in Argus the names are different, but the information you need to provide is the same only what you need to provide in ARISG as well. So the Argus, the name of the tabs are general, patient, product, event, analysis, activities, additional information, and regulatory reports. So the names of the tabs are a bit tricky. So in ARISG the tabs are easy to understand, basically the names are easy to understand. So some of the tabs names are self-explanatory. So in general tabs we just do nothing, just provide the information of the reporter, the person who is reporting. So basically there are two reporters, primary reporter and secondary reporter in pharmacovigilance. In primary reporter, the person who just directly reports to the marketing authorization holder, who directly calls or reports to the particular company, is known as the primary reporter. On the secondary reporter is the person who is not directly reporting. For example, it may happen that a patient is reporting to his physician and the physician is calling or physician is mailing to the pharmacovigilance department of a particular company. So in this scenario, the primary reporter is physician, that particular physician, because he is directly reporting to the company, and the secondary reporter is that patient who has informed that physician. So patient, if directly reports to the company, then patient will be the primary reporter. So in general type, we just provide the reporter details. We just fill up those name, address, telephone number, etc. of the reporter. Then in patient tab, we just fill up the details of the patient. Details of the patient means we just need patient demographics: age, sex, race, ethnicity, pregnancy status, date of birth. These all things we need to provide. Then in under patient tab, there is also another subtab known as disease subtabs, where you need to provide the disease condition which a patient is undergoing or which a patient has experienced before taking our particular company suspect drug. So particularly company suspect drug means the drug which is suspected to produce event. For example, I am telling you an example. For example, I have taken a drug X, and after taking drug X, I have experienced headache. So I am just reporting it to the company that after taking your drug X, I have experienced headache. So before taking a particular drug X, what all conditions we have experienced, or I have experienced, or the patient has experienced, or what all drugs that patient has taken before that taking that particular drug, those are considered as history. Those things are filled up in the disease subtab which is present in the patient tab. So those particular information, the disease, for example, patient may develop some problems during his birth. So any sort of diseases starting from birth till taking a particular company suspect product, all those things will, all those disease conditions will be need to be database or need to be reported under the disease subtab which is present in the patient tab. Then comes product tab. In product tab, we need to fill up the product details. So first in this, we need to identify the company suspect product. The company suspect product, I have already provided the example. Then we may sometimes need to fill up the concomitant medication details. That means what all sort of drugs patient was taking along with that particular company suspect drug which has suspected to cause the event. So for example, I was taking drug Y and Z as well while taking drug X, but I am suspecting drug X is causing event because after taking drug X I have experienced these events. But at the same time, drug Y and drug Z was also, I was also consuming drug Y and drug Z. Maybe I have started it before taking drug X, but maybe after taking drug X, but I am taking it along with drug X. Conmed means which I am taking it with along with the suspect drug, but the drug may have been started before taking that particular suspect drug, or may have been started after taking the particular suspect drug, but it should continue along with that particular suspect drug. So this is the idea of conmed. This is known as concomitant medications, or conmed in short. So another is drug used to treat AE or adverse event. So for example, we need to fill that as well. So what is that? For example, after I am taking drug X and I have experienced particular event, and to treat that particular event, my physician or I have taken a particular drug. So that particular drug is known as drug used to treat AE. It is also like concomitant medications only, but it is specifically drug used to treat AE. That means after taking adverse event, to prevent or to eradicate that adverse event, I have taken a particular drug. So that is known as drug used to treat AE. So we need to fill in this way, need to fill different drug details, then for what indication we have taken that particular drug, when we have taken that particular drug, what was the form of administration, what was the route of administration, where it was administered, what was the lot number, the batch number of the product which we have taken. So it is generally available in the packet from which you are taking the particular drug, or those, even if you don't have the information, then leave it. You can also keep those details blank. So event information, this is the critical task in pharmacovigilance. We need to identify the event, what patient has experienced, and we need to database those events in the particular event tab. So as I have already said, patient will use different layman languages, and even foreign country people, they use foreign English. We know that foreign English is something different. It's a bit difficult to understand, and their layman language is something, sometimes it becomes very difficult to understand those English language as well. So we need to understand those layman languages, and we just need to fill up those details in the database, and we need to identify the particular medical concept associated with that particular event. So the example which I was telling, the pain was so severe, it seemed that someone was hammering on my head. So in this case, only the event pain is directly clear, that patient is experiencing pain. But if we just closely monitor the sentence, we will see that it is associated with head, because patient has said that someone is hammering on my head. That means definitely it is head pain, which means headache. So this is some sort of headache. Now again, we need to identify whether some more medical concept is available here or not, or some specific medical concept. So more specific concept, try to identify the best specific concept. So someone was hammering on my head. So hammering, if you just see or Google out the English term, so hammering is somewhat, it is somewhat like throbbing. So you might have heard about the throbbing headache. The term throbbing means it is somewhat like throbbing headache. So the concept of throbbing headache we'll use to quote. So the coding of this particular event, the exact medical concept of this particular event is throbbing headache. So if you just, on the sake of data, if you just blindly code it with pain or head pain, then it will not be perfect, or it will not be appropriate. There will be error. You have to code it with throbbing headache only. So in this way, you need to identify the medical concept. Next comes the narrative part. In narrative part, we all know, so narrative is basically, there exists a format, there exists a template for writing the narrative, and nowadays most of the things are automated. So narratives are automatically written in most of the databases. Companies have developed lot of tools, and in those tools narratives are auto-generated. So narratives got automatically written, and narrative is something what we have written in our school life. So we all know how to write narrative basically. So the idea is also basically same here. There is some basic template is there, we need to write accordingly, but you need to just arrange those things properly. Means you need to be good in English, or you need to just add those things in a proper English, so that it looks beautiful, or everything is written in a synchronized way. The chronology of narrative, this term we often use in pharmacovigilance, chronology of narrative. This chronology is very important. We have to write everything in a chronological way, in proper synchronized way. Especially we need to discuss the event related information, what event the patient has experienced. Patient, they will also just provide, they will report those things in the form of a narrative only. Patients, they will just provide those information in the form of paragraph, but we need to arrange those information in a proper synchronized way,
imparting or using medical, proper scientific medical terms. So this is the concept of narrative writing. And regarding causality or leveling, there is a dedicated slide. I will just explain you those things in those slide. And regarding lab, so regarding lab, as the name suggests, what all lab patient has test means being tested or undergone through. So example, what all lab test or diagnostic test patient has undergone through while when patient has experienced event, and for that event what all labs the patient has undergone basically, or sometimes what all labs patient has undergone before taking or before experiencing that event. Those lab details may also be required to be filled up. So patient, they will just provide all those information in their ICSR form, that particular form, and based upon this we just need to fill up those necessary details in the laptop. And again, here also patients are using layman languages, or sometimes they will just use some terms which will be non-medical or non-scientific. So you need to quote that particular term as well. You need to identify the exact medical concept associated with particular lab, and you need to code it. So again, here you need to apply your brain and the knowledge of anatomy and physiology. And then there comes study. So basically study, these also I will just explain you in some other slides. I have also information about this. And other database, Argus. This Argus database is often more used compared to RSG, because this Argus is well advanced, but the name of the tabs are little bit difficult actually, but the things are basically same. The advantage of Argus over RSG is that there are less number of tabs, and it is more customer user friendly. So people are just shifting, or the companies are shifting to Argus basically. But again, you need to fill these details only under these Argus tabs as well. So this is the basic idea, basic concept, what we just do in our pharmacovigilance field. After getting a job or after getting a pharmacovigilance, what you need to do basically, I have provided you the basic details. So next slide please. So no, previous slide. Hello Shobhata. Yes, this one. So where from we are receiving those reports basically? So we are receiving ICSR reports from solicited source as well as unsolicited source. So source of report, this is also an important question for pharmacovigilance. So solicited means the report, as the name suggests, it is received from an organized source. Means there are certain people, certain sort of people which are being hired by a company, or there are certain people who have certain sort of obligations for reporting a particular adverse event. Means their job is to report adverse event only. So it is mandatory for them to report those particular adverse event to a particular company. That means company are hiring those people for reporting those adverse events only, or company have some contract with that particular organization, so that they will just provide or collect the report from the patient parties, and they just will provide the details to the associated marketing authorization holder. So those reports are known as solicited reports, which are derived from organized data collection system. So examples are clinical trials, patient support programs, different patient surveys are conducted in US, etc., etc. So these are some organized solicited sources. So these are basically based upon studies. Basically for solicited cases, we just need to fill up the study information, which I have shown you in the previous slide. There is a tab called study tab. So if your report is solicited, then it must have come to a certain organized study program. So this study program, we need to fill up the details of that particular study program. We need to select the pop-up, there are a lot of pop-up available, and we need to select the particular study, name of the particular study program from which we have received the report. So this is this idea of solicited report, and unsolicited report is a spontaneous report. These people, these reports, there is no such obligations or no such mandatory thing that that report needs to be reported. There is nothing mandated. Means it is your choice whether you will report or not. So, for example, patient, physician, healthcare professionals, those who are prescribing, or the person who is taking the drug, so they can just take report if they want, they wish, or if they do not want to report, then there is no harm. The company will not just come to you or disturb you to get that particular adverse event report. So it is your choice whether you will report or not, but it is always instructed or suggested to report. So these are certain spontaneous report. That means there are no certain obligations for reporting. You may report or not report. You must see the report randomly. So these are known as unsolicited reports. So examples, some other examples are literature reports. Even you must have seen different articles, publications are there. So there are certain publications related to pharmacovigilance or as well, and there are a lot of journals published, and different people conduct different study or different analysis related to post-marketing drugs. So what sort of experience they have seen in particular group of patient who are under these conditions. So based upon this, different review articles or different research articles are published. So marketing authorization holders, they need to track all those articles, maybe on weekly basis or twice a week, and those are also considered as a source of report. This literature reporting or literature case processing, we mainly call literature case processing, is a very noble profession and very important. You will learn a lot of things if you are into literature case processing, and just people, they just want to go to this literature case processing only because this is one of the unexplored and very noble profession in the field of pharmacovigilance, and it is tricky as well. You need to apply a lot of scientific as well as medical judgment as well as lot of knowledge as well, because you must have seen the articles are very big. Any research article is not that simple, it seems to be boring. So it is very critical as well as novel and as well as prestigious. So there, even internet also considered as a source of reporting. Press, media, regulatory authorities, they also serve as a source of reporting in the field of pharmacovigilance. Even regulatory authorities, as a person, even if I am a patient and if I am just reporting to the regulatory authority, I am also allowed to directly report the case to regulatory authority. It is not like that it will go through the marketing authorization holder only. It may directly go to the regulatory authority directly. So I can directly report it to the regulatory authority, and regulatory authority in turn, they will report it to the marketing authorization holder. So this is also possible. So next slide please. So I will just wrap up. I'll just make the video short. Basically it's taking a lot of time. So basically this is first of all valid ICSR. Once we are just receiving a report, we need to just see whether our report is valid or not. So we just basically look for whether our report has an identifiable patient. That means that we have a particular patient with that particular demographics. For example, if a report is received randomly and we don't know any sort of demographics of the patient, then that report is known as unidentifiable. So a report should have an identifiable patient. That means a patient information with particular demographics of the patient should be available. Then there should be a reporter details. So particular reporter details, identifiable reporter. So basically sometimes it may happen, especially for internet or different company has their own Twitter account or social media pages. So sometimes it may happen to blame a company or to just to make a harm to a company or just to harm the reputation of a company. Some people, they just produce a fake report. They just create a fake ID of themselves and they just keep a fake report They post a fake report. They just blame a company. So this is a one important thing, that a reporter who is reporting a particular case should be identifiable. The report should come from a particular, if it is coming for a social media page, it should be come for a genuine profile basically. So in Facebook, we have seen that in Facebook also some of the profiles are fake basically. So in pharmacovigilance also this problem is there. People create fake profile and they just try to harm the reputation of the company. So reporter has to be also be identifiable. Then company suspect. So basically, if there is no company suspect, for example, after taking a drug of particular X company, if you are experiencing an event, but if that particular X company drug is not there, then definitely there is no means of reporting. So particular company suspect is also important. At the last criteria is valid event. Valid event, that means patient must experience a particular adverse event or any issue with a particular product after taking that particular drug. That is known as valid event. So at least one event or particular issue should be present for reporting ICSR. So this is the concept of valid ICSR. So if any one of the four is missing, we will collect the report. The person will definitely, the marketing authorization will collect the report, we will acknowledge the report, we will just enter in our database, we will just get a case number. Case number means it is equivalent to the application number or the registration number of which is associated to a particular reporter, the particular patient who is experienced the event. We will just record it. It will keep it on database, but this report will not consider for regulatory authority submission. That means we will just keep it for our documentation only. So it will be databased and we will just wait for a follow-up information. We can follow up, we can just contact the person who has reported. If he or she definitely urges or shows interest, if she provides the consent to contact, we will definitely contact in future to collect the extra information about that particular report, the missing information. And until and unless the report, any one of the four criteria we are just getting, so any one of the four criteria is missing, until and unless those criteria we are getting will fulfilling all the four criteria, we will not consider this case for regulatory authority submission. Regulatory authority will not consider this report. They will not accept, they will simply reject it. So next comes the timelines of submissions. Since it is healthcare industry and it is concerned with the health of the patient, so timeline is very important. So a report needs to be submitted to the regulatory authorities within a stipulated time only. So this timeline, again, this is very important question for pharmacovigilance interview. This is in fact a patented question. Especially they ask this question to the experienced candidates, but if your luck is bad, they may also ask to the fresher as well. So timeline for reporting pharmacovigilance. So if the case is death or life-threatening case, that means the case, any particular adverse event has led to the death or it has caused a serious life-threatening problem to a patient, to the victim, then we need to report the case within 7 calendar days to the regulatory authority. That means this case needs to be submitted to the regulatory authorities within 7 calendar days, provided that the report is valid. These 4 ICSR criteria is meeting. We need to submit within 7 calendar days for clinical trial cases. That means if the product has not been introduced in the market and if the product is on phase 1, phase 2 or phase 3 clinical trial, then that should be reported to the regulatory authority within 7 calendar days. And if the event has caused death or life-threatening incident and if the product is already in the market, then the time frame of reporting is 15 calendar days. That means this report should be submitted to the regulatory authority within 15 calendar days. And also note that death and life-threatening cases, these are always serious and these are considered to be unexpected. Generally, these are unexpected. There are certain exceptions, but very rare, because a company will not develop a product or deliberately develop a product which will lead to the death or life-threatening incidents. So a company will never just consider or just try to develop a product with this sort of adverse event. They will definitely try to minimize this sort of critical adverse event, but in certain instances, death or life-threatening conditions may occur with a particular product and it may be expected, but mainly in 99% of cases these are unexpected, and those death and life-threatening incidents needs to be reported within 7 calendar days for clinical trial cases and 15 calendar days for post-marketing cases to the regulatory authorities. And other serious cases, so there are various seriousness criteria which I will just take you through the next slide. So for other serious cases, the regulatory timeline is 15 calendar days for both clinical trial and post-marketing cases. Again, these should be unexpected. So any unexpected serious cases needs to be reported through expedited reporting within 7 or 15 calendar days for death and life-threatening cases, or for other serious cases the timeline is 15 calendar days. And unexpected, everything should be unexpected. And if it is expected serious events or any unexpected non-serious events, these are generally reported through aggregate reporting, which aggregate reporting concept I have already explained before. Aggregate reporting is basically nothing to just report those things in collated format. That means in periodically after a while, within short period of time. So means maybe in 1 year or after 6 months. So non-serious cases or unexpected serious events are reported through aggregated reporting. But in certain countries there is still some local regulations. For example, in European countries they ask to report non-serious cases within 90 calendar days. So the time frame is very huge basically. So again I am saying this timelines is important, very important, another patented question in the pharmacovigilance, especially for the serious cases. The timeline you need to remember 7 or 15 calendar days, 7 calendar days for clinical trial cases, 15 calendar days for death and life-threatening cases, and for the other serious cases it is 15 calendar days for both clinical trial and post-marketing cases. So next slide please. So what are those seriousness criteria? So this you may have read or may not have read in pharmacology, I'm not sure. Basically, an adverse event is considered to be serious based upon its outcome basically. So if the adverse event has resulted in death, it is life-threatening, or if an adverse event requires inpatient hospitalization or prolongation of existing hospitalization. So prolongation of existing hospitalization is something that, for example, patient was hospitalized due to some sort of infection or sepsis. Now what happens basically, he is in hospital for 3 to 4 days. Now what happens after 3 to 4 days, it was invented that patient was about to be discharged, but it was found that patient has developed or acquired some other condition, or sometimes the condition for which the patient has been hospitalized has worsened, and due to which the time period of hospitalization, for example, the time period was initially 3 to 4 days. It was expected that the patient will get out of the hospital after 3 to 4 days. But due to some unwanted incident or any sort of event, the hospitalization of the particular patient is getting prolonged, or it is getting increased. The duration is getting increased. So within an existing time frame of a particular hospitalization, if the patient is again having some sort of problem due to which his hospitalization stay is getting increased, the duration is getting increased, or if the patient is again re-hospitalized for a particular event. For example, patient was admitted for sepsis, he was discharged after 4 days staying in hospitalization, and after coming back to home again he just went to the hospital with the very next day, on the very next day, for that same problem. So what happens? The hospitalization duration has increased. So whatever doctor had identified, doctor based upon study, doctor said that the patient will be discharged within 4 days, but due to some reason, patient may require to get rehospitalized, or his hospitalization duration has been prolonged. So this is the idea of prolonged hospitalization. So this concept of hospitalization and prolonged hospitalization is different basically. So another thing is results in persistent or significant disability on incapacity. So any sort of persistent disability means, persistent or significant disability on incapacity means there is a substantial disruption of a person's ability to carry out normal life functions. So for example, I will just say what is persistent and significant disability. For example, you are not able to do your daily activities due to experiencing some sort of disability. For example, you are paralyzed, or your limb gets amputated, or you may develop some sort of problems for which you may not carry out your daily activities, some sort of daily activities, or you may become physically challenged. So these sort of problems are basically considered as events which are resulting in persistent or significant disability. So this is the idea of persistent or significant disability. And any event which is causing congenital anomaly or birth defect. So basically, if an event is causing birth defects, that means the example is phocomelia, which was found in case of thalidomide disaster, or when a baby is in the womb of a patient, and the female patient, if she takes a particular drug, and which may cause certain sort of anomaly or congenital teratogenic defect in that particular children, and due to this the baby is born with some sort of permanent defect. So lot of defects are there, like trisomy, then different sort of other defects. So those defects and which are present acquired, or which are not acquired but present since birth, that means congenital, developed due to mother's exposure to a particular drug. So those sort defects are known as congenital anomaly or birth defect. And IME, important medical event or medically significant event. So this is some sort of miscellaneous. So the events which are not fulfilling the above criteria, but again these are considered to be serious. For example, I am saying, so there is some sort of IMA list which EMSI, that means European Medicine Agency, they periodically, twice in a year, they just publish some slides, a list in which there are some sort of events which does not fall under this 5 criteria, but those events are still considered to be serious based upon their criticality only. So companies, they download those particular list from that EMA site. Even you can also download if you go to the ema.gov.org. So if you go to this site, then you will be able to see that IMA list. There is something known as IMA list. You will see the list of those events. So those events are considered to be serious. That means miscellaneous types. For example, I am saying one event: cancer, stroke. So cancer is something which is not fulfilling under these criteria, any of the criteria. So this cancer is considered to be a medically significant, because cancer, we know cancer is a very serious event. It may also lead to death. or for example AIDS is also there, then myocardial arrest, heart attack, these sort of events. It's not fulfilling under any sort of criteria, but these are equally serious. So these all sort of events are considered to be IME. So these are the various seriousness criteria, and again this is one of the important question. They will not ask you to explain the seriousness criteria, but they will just, in interview they will just ask to just list the name of the seriousness criteria. What are those all the seriousness criteria which are present in pharmacovigilance, which pharmacovigilance deals with. So these are all the seriousness criteria in the field of pharmacovigilance which a case may have or which a patient may experience after taking a particular drug. Next slide please. So coming to the concept of MEDRA, so again I will just, MEDRA is a huge topic, so if you want to get a detailed idea you can visit our channel. So the link is present in the description only. So basically I will just brief you regarding MEDRA, because from MEDRA also you will be getting questions in interview. So basically MEDRA is nothing but, I was saying that the people they uses the various layman languages, they use very unscientific languages, and we need to identify the medical concept what is available, and we need to code that particular medical concept only. So it is somewhat like your barcode. Basically in your OMR sheet you will see that the barcode is present, or any in barcode there will be lot of, there will be lot of digits. So each code is provided with a particular particular digit basically. So same way there are some event terms, for example fever, headache, this. So these codings are already available in the database, in the MEDRA database, and these are particular 8-digit numbers, and each event is provided with the particular 8-digit number which is unique to that particular event only. So to represent a particular condition, particular physiological condition of a patient with a particular medical concept, MEDRA is used. So MEDRA is a clinically, the full form is present here, Medical Dictionary for Regulatory Activities, is a clinically validated international medical terminology used by regulatory authorities and the regulated biopharmaceutical industry. So it was developed by ICH as MS. So ICH, International Council of Harmonization, you may know the full form of technical requirements for the regulation of pharmaceuticals and in human use. So this is the full form. Then MSSO, Maintenance and Support Service Organization, it was developed by these two organizations and is responsible for maintenance. So these are some sort of terminologies which should be used by the regulatory authorities. As I have already said, regulatory authorities, they will not accept those layman languages. They will accept those particular medical concept associated with particular layman language or the particular disease or physiological condition. So that we just need to come through, or just we need to code or to identify using MEDRA. So medical conditions like indications, investigation, that means lab test results, medical and surgical procedures, social and family medications, errors, product quality issues, that means issues associated with the tampering, counterfeit product, or any sort of quality defects, device related issues, product use issues, pharmacogenetic terms, toxicology issues under the scope of MEDRA. So there are a lot of things, that means not only adverse event but lot of other things can also be coded through MEDRA. So again, this is very important update of MEDRA. So it is very important question for interview purpose. The update is provided, update is done in the month of March, in the March and September every year. So these updates are done biannually basically, so twice in a year after every 6 months. So current version is 25.50. In September it will be 25.1. So So major update. So Medra has basically two updates: major update and minor update. So major update is done in the month of March, and in minor updates done in the month of September. So major up. So yeah, that's it. That is sufficient for now. And in for hierarchy, if you consider, Medra has 5 hierarchy. So if an event is there, for example flu, so hierarchy is basically nothing. I'm telling that hierarchy is group of concept. So you must have heard the concept of species, genus, family, organ, class, phylum, kingdom for animal system of classification. This concept you have you have heard of. So in our school life in biology, this was there. So group of species will consider genus, group of genus will make a family, group of genus family, then comes order, then group of order, then in this way it will go till kingdom. So group of small terms will make consider will just form the bigger term. So group of LLT will just form to be PT, group of PT will form HLT, group of HLT it will form HLGT, and group of HLGT it will form SOC. So there are 5 stages of hierarchies. So for example, I have also provided one example. If LLT is flu, then it is flu is a type of influenza only, and influenza is a type of influenza viral infection only, and influenza viral infection is a type of viral infectious disorders, and viral infection disorders is a type of infections and infestations. So in this way it comes, and while coding you need to identify this LLT only. You are not required to bother about this all the rest 4 terms. So while coding, if you just type flu, then you will get the idea, basic concept. You will also get this all these 4 things. So what is the key in SOC, HLT, HLGT or PT available with that particular event flu, you will get all the things will get auto coded. So you need to code flu only. So next slide please. So I have just highlighted some of the examples I have of event verbatim and coding. So what event verbatims we just receive. So symptoms, if event verbatim is a symptom, so it is say that patient states she has been experiencing cold sweat. For example, this is the narrative, this is the information which I have received in our narrative. Patient states she has been experiencing cold sweat. So cold sweat is basically a symptom. So if we search the Medra browser, we'll identify one low level term or LLT called cold sweat. We need to code it with that. So this one is very simple basically. Some of the events are basically simple, but some of the events are complex as well. Another one, the second is investigation. Lab test result indicates that the patient has increased troponin and increased CPK-MB. So how to code this one? So increased troponin, we need to search for this term troponin, and when we just type troponin in the browser, we'll receive lot of terms associated with troponin. Troponin increase, troponin decrease, troponin abnormal, troponin issue. In this a lot of things we'll just find, and we need to select troponin increased from there. So, and the SOC of lab test will be always investigation. In the similar way, there is something likewise we can quote CPK-MB increased as well. So regarding patient demographics, a 2-year-old baby was noted to have a mild fever. So mild fever, it is basically, if we code it with mild fever, then if you can code it with fever, but this concept will be wrong, because here we need to capture the verbatim, the event verbatim we need to capture in our database as 2-day-old baby developed mild fever. This 2-day-old baby is important, because if we just type fever in Medra browser, we'll get lot of concepts, and one is fever neonatal. So 2-day-old baby, it means neonatal. So the ideal concept will be fever neonatal, not normal fever. So we need to code in that way. So you need to consider lot of things before coding event verbatim, and at the same time while taking or extracting out the particular event verbatim from a narrative. So what all information we need to capture or we will not need to capture, we need to have a clear idea on this. We have to need to have a thorough idea. So it is not that much simple basically as people think. We need to identify the basic medical concept, and for this thorough knowledge of anatomy and physiology is important. So indication: a 35-year-old woman was taking drug X to prevent relapse of multiple sclerosis. So if we take multiple sclerosis only, then it will be coded to multiple sclerosis as an event. Now my purpose is to code an indication. Purpose is to prevent relapse of multiple sclerosis. So we need to code it as multiple sclerosis relapse prophylaxis. Next is product quality issues. Product was found to be counterfeit. So product counterfeit is available. Social circumstances: the patient was confined to a wheelchair. Even if you are using a wheelchair, this is also an event. Wheelchair user. Or if you are even using a glass, you will also get I8 user, there is a coding. And if you are using in a walking aid or walking on clutches, then there is a code available for walker aid user. So in this way, these social circumstances are also codable. So pharmacist had made a mistake in compounding the medications, that is medication error, product compounding error. This code is available. So you need to type this term compound, and when you type this term compound, you will get lot of events basically, and you need to select the product compounding error. And there are even some vague terms which are sometimes very difficult to identify. What is this basically? So hypotension turned green. So these are basically pain. Hypotension is nothing. It seems to be there is some, it should be hypotension, but there might be some typo error or spelling mistakes. So hypotension is not a medical concept. You need to identify and code accordingly. And turned green. So turned green is something like your skin may turn green, or you may face some sort of skin related problem or some problem associated with the physiology of body, or even a tablet or any sort of medicinal product, the appearance of a product can also be altered. It may become green. So the exact issue is not clear. What is getting turned green? Whether it is a disease or it is a product issue, or what is the particular issue. So this is such concepts which are totally vague, which cannot be identified, which cannot be coded. Those are coded with the term unevaluable event. So next slide please. So now I will just provide you a just very little idea about causality and leveling. So causality, as I have already said, it deals with finding a temporal causal association between a drug and a drug reaction. It is an evaluation of a likelihood that a particular treatment is in cause of an observed adverse event or not. So what happens basically after taking a drug, you have identified a particular event. So you may think that the drug, that particular my suspect drug has caused this event. Since after taking this drug, I have experienced this event, so my drug is responsible for causing the event. But the scenario is not like that. There are a lot of other conditions. A patient may have lot of comorbidities. Patient may take lot of other medications. Patients usually take pile of medication. For example, you are taking drug X, Y, Z, blah blah blah, and a lot of other drugs. So you may, as a non-healthcare professional, as a patient, may blame drug X. But the thing is that drug Y and Z may equally be responsible for causing a particular event. So how to identify, how to know that? So that journey is known as causality assessment. So while reporting, it is always always intended or always desired that a patient must report everything in clear detail. Means any history, medical conditions, concomitant medications, any sort of family history, genetic disorder, everything it is expected to report. So the person, the concerned person of pharmacovigilance, they ask all this information to the specific patient, and it is always encouraged that you report everything, as causality assessment will be based upon those informations only. So the so causality depends upon the time relationship between the drug and the adverse event, the competing causes, response to a drug withdrawal and dose reduction, response to a drug administration. So there are a lot of factors in this way, and expectedness is means leveling, where we determine if the event term is already included in the reference safety informations, as I have already said before. So to determine whether the nature, severity, specificity and the outcome of a particular event is in accordance with the one present in the reference safety information or not. So there is those patented or that particular dictionary, or you can say that standard document is there, and from standard document we just need to search whether the particular event, the particular coded event, for example the event fever, is present in that particular document or not. We can just press Ctrl F, we can just open the document, we can press Ctrl plus F, and we can just search that particular event, whether it is present or available or not. If it is available, then it is well and good, and if it is not available, then you will consider this as unexpected. But there is another twist here also. We need to identify the nature, severity, specificity and outcome of that particular event. So it's not like that we'll just search Ctrl F and we'll just find that event is present anywhere in the document, so this event is considered to be expected. It's not like that. The nature, severity, specificity and outcome needs also to be considered. For example, and there are certain sections in that particular reference safety information from which only we need to select that particular, from which we just need to find whether this information is present in the particular section or not. If in those section, if that particular information or the particular event is present, then only we will consider this event has to be labeled. So we just look after the contraindication section, warning, precautions, adverse event sections, and in this way there are some other sections as well from which we need to observe or need to find. So some of the examples I have shown here for this nature, severity, specificity and outcome. So prolonged elevated liver function test is considered unexpected when leveling states only transient elevated liver function test. So see, prolonged elevated liver function test means it is a chronic condition, and transient means it is an acute condition. So if something is acute, some acute condition is present, but if you are experiencing an event or something in a chronic basis, then you need to consider that chronic one as unexpected. Again, circulatory collapse is considered unexpected when hypotension is leveled. So circulatory collapse is something which is more severe form of hypotension. So there is continuous fall in blood pressure and a prolonged and very severe form of hypotension. When there is a severe fall in blood pressure, it may lead to circulatory collapse, and if your event is circulatory collapse, then on basis of hypotension you will not consider circulatory collapse as leveled. Left-sided chest pain is equivalent to chest pain. It should be assessed as unexpected if chest pain is expected. So specificity, what happens? So considering anatomical location under this specificity, so if you are experiencing left-sided chest pain, you have reported that I have experiencing left-sided chest pain, but your leveling document states that I have chest pain. So left-sided chest pain is considered, we can just consider equivalent to chest pain only, because the medical concept will not change, only the location is altering. So if chest pain is leveled, then left sided chest pain will be considered as leveled. Next, regarding outcome, if pneumonia is expected but the patient experienced fatal pneumonia, the fatal pneumonia should be unexpected. Fatal pneumonia means pneumonia leading to death. Document has stated that patient has pneumonia, but patient has experienced pneumonia and the patient has died. So definitely document doesn't state that patient will die out of pneumonia. The idea is that in that sense, pneumonia will considered to be, fatal pneumonia considered to be unexpected. The event will be coded to pneumonia, but the outcome of the event will be fatal only, and hence the event will be considered to be unexpected. So you need to consider all these things while judging leveling as well, just searching for information whether the information is, whether the particular event is leveled or not. So you need to consider everything: nature, severity, specificity and outcome. Just doing control F and searching for a information for a leveling is not enough. So, and leveling is very important, as I have already said, because based upon leveling only the reportability of the report of a particular adverse ICSR report will depend. Expedited reporting, non-expedited reporting, so the case will be submitted or not to the regulatory authority, it will be based upon the leveling only. So we need to be careful while doing the leveling as well. Next slide. So now we have just towards the end of the presentation. Basically it has been around 2 hours. So now I'll just provide you the what are the interview questions which you can expect as a fresher if you want to just join pharmacovigilance field, which I have experienced or Shogata has experienced or others person have experienced. So basically the interview question, basically first of all I will just tell you what all things will make you upper hand, or will just, they are just look for, they just prefer if you want to seek job in pharmacovigilance. So first of all, you will always be preferred, or it will be upper hand for you if you have a good knowledge of anatomy and physiology, if you have good in English, and if you are good in Excel of computer. So these three things are important to be a pharmacovigilance, to join pharmacovigilance course or to be in pharmacovigilance, because you need to deal with clients, you need to be well good in writing English, especially mail etiquettes. You need to deal with lot of mails, you need to handle a mailbox, you will receive plenty of mails, sometimes it will be around 60 to 70 mails per day. You need to be expert in handling mails. So they will also provide you training, but you should know how to handle the mailbox, how to segregate the mailbox, how to flag the mailbox. So you should have that idea, or you should have you should know how to write a email basically. So professional email etiquette is very important for pharmacovigilance. And if you can speak in English, if your English is good, if your grammars are good, then definitely it will be welcome, because these are already US based companies basically, and they will use English only. You may need to converse with clients directly, you need to talk with clients, there will be a lot of client meetings, need to handle the clients. So definitely, and their English, you know that US English is not equivalent to Indian English, their pronunciations are different. So you need to be good in English, or at least after joining, there are in IT sector, they provide you lot of courses, short-term courses free of cost. You can take any of the courses and you can develop your soft skill in any of the ways. So if you are not good in English or not good in Excel, then don't lose confidence. You can join, and there will be lot of courses free of cost. where you can enroll yourself, and even your team or even in IT sector, people always encourage you to just join and to develop your soft skill. They always encourage you to join those courses or just participate on these courses to develop your soft skill. So basically Excel, then English and anatomy physiology. As I have said, this MEDRA coding, it is totally based upon your anatomy physiology knowledge. So the more you read anatomy physiology, the more you love anatomy physiology, the more it will be your upper hand. So if you are not good in anatomy physiology, then also don't worry. Again, there are a lot of courses available. You can, after joining also, you will just get to know lot of things, and again there will be lot of people to help you, because in pharmacovigilance, this the professional sector is like that. You will have plenty of doubts, and number of doubts per day. You may have 40 to even 20 to 30 doubts per day, or even more than that. But there are a lot of people available on floor. Their job is to solve doubts only. So there will be lot of trainers, lot of their colleagues. They will definitely solve your doubt. They are they are very much friendly. They will always solve your doubt. So don't lose hope, and don't hesitate to ask doubts. So now moving on to the interview questions. What questions they basically ask? First of all, what is pharmacovigilance? Phases of clinical trial, adverse drug reaction and adverse event, basic differences. These three are basic patented questions which you will almost 99.99% you will expect as a fresher. Then what is MEDRA? I have already provided current version of MEDRA hierarchy, that hierarchy which I have said, HLT, LLT, then SOC, that hierarchy. Some basic definition, this you will find in internet. Medication errors, there are various sort of medication errors: off-label use, misuse, abuse, lack of effect, product quality complaint, overdose, underdose. These some of the definitions you may have seen in pharmacologies, and some of the definitions are available in internet. Upon browsing you will get these definitions. Then narrative writing. This narrative writing, they will just ask you to just write a narrative. They will just provide an article, patient information, details about adverse event details which a patient has experienced, and they will just see how we are writing the narrative. So don't worry, the narrative, through this they will just see your English skill. Actually basically you are not required to follow the strict template which companies do follow while writing narrative. They will just basically see your English skill and the way, the chronology, the synchronization, how you are writing the sentences. So the idea is basically like that. Then reporting timelines, the 17 calendar days, 15 days, 90 days, which I have already said, that is again a very important question. Then seriousness criteria, those seriousness criteria, life-threatening hospitalization, IME, those criteria, the name of those criteria are important. Then valid ICSR and invalid ICSR, those four criteria: identifiable patient, event, suspect, reporter. These are valid criteria, and invalid, if any one of the above all those are missing, then it is considered as invalid. So they will just ask this one. And day zero or IRT, this I have not said, I am just saying it briefly. So whenever a report is received, whenever a patient is reporting to a particular marketing authorization holder or any other people, basically regulatory authorities, or any Facebook post is being posted. So whenever a company is basically getting aware of the product, of a particular report, particular valid report, definitely valid report means those four criteria wala report, they are getting aware of that for particular criteria wala report. The day on which they are getting aware of that report, that is known as day zero. So it is not like that, for example, a patient is reporting on Sunday and no one was there to receive that report. Patient has dropped a mail. Now on Monday the office opens and the people will find that patient has reported the report on Sunday. So it is not like that the report will, day 0 will be considered on that Monday only, but the day 0 will already be considered for starting from Sunday, because the patient has reported that particular valid report on that particular Sunday only. They have already posted or mailed the report on that particular off day. So the day 0 concept is independent of business days. It is based upon calendar day only. So the day on which a report is being reported is known as day 0 or IRD, initial received date. The term is important, initial received date. So the various health authorities across various countries, so there are various regulatory authorities. The name of these regulatory authorities are already, you can get it in internet if you browse, or even we have a dedicated video in our channel where you will find the country-wise regulatory authorities name. You can visit our channel as well. So for example, I am saying for the health authority of USA is FDA, or health authority for European country is European Medicine Agency. For India it is CDSCO. So in this way different health authorities are there country wise. So some of the country wise health authority names you try to remember. Country like European Union countries, then USA, Japan, then India, then some neighborhood countries. Try to remember, memorize the name of the regulatory authorities of particular countries. Sometimes they may ask questions on this. But again I am saying these first 3 questions, pharmacovigilance, phases of clinical trial, adverse drug reaction, adverse event, these 3 are patented questions. And apart from this, they will just ask you about your some knowledge about some relevant topics on pharmacovigilance or some topics related to pharmacology, basic pharmacology, or many mechanism of action of a drug, any classification, these things they can also ask you. So next slide please. So these are the list of some of the companies, top companies where with just pharmacovigilance is available. So out of these first 2 companies, Cognizant Technology Solutions and Wipro, is only in Kolkata, and rest of the companies are basically in South India or West India. So people of Kolkata, we basically start off our careers with these companies only. Even I have also started with Cognizant only, but after that they just switched to various other companies. So mainly people, I have seen that the trend is that people have a very high fever of switching to these companies, IQVIA, Biotronica, LabCorp, and Tata Consultancy Services also they just preferred. And apart from this, there are lot of other companies as well: Accenture, Syneos, Novartis, Novo Nordics, Eli Lilly, Paraxel. So these are some sort of companies. They will also provide package as per your current package. So for your career growth, you will definitely just try to switch to some other companies as well. And if you try to switch to other companies, then some companies they also provide permanent work from home facilities as well, but the seats are very limited basically. Some of the companies are there, for example this IQVIA, Syneos, they just provide permanent work from home, but the seats are again limited. But as a fresher you will not be getting a permanent work from home for any of the companies. Even each and every company has permanent work from home policies, but this IQVIA, Syneos, etc., these have the number of quotas are higher. So again, you need to search and you need to browse and you need to see where you just need to just relocate, or whether you are interested to relocate outside or not. And the thing is that if you just make one or two switches in pharmacovigilance, the sector is that your career growth will be such high, because the package which you will get after one or two switches will get a tremendous hike, and after that you will be confidently able to settle outside with your family even, because the package they will provide is tremendous. Since these companies are already being influenced by foreign policies, and these are IT based companies, so after staying for a while, for a period of 4 to 5 years in this sector, so your package will increase at a very good rate. You will get a very good package. So if you wish to just be in this sector, you can be in this sector. The thing is that there will be no physical effort, but yes, some mental efforts may be there. Work pressure sometimes is high, and you will get good salary. The thing is that ultimate thing is that you will get a good package basically compared to QC or production. Next slide, please. Yeah, that's it. Thanks for tolerating me for 2 long hours. I don't know whether I have bored you or not, or whether I was able to explain the things or not correctly, whether you have understood anything or not. But anyways, it was a nice presentation, and I was thankful to you for joining this presentation. And this topic is basically very vast, and it is very difficult to cover everything in a short period of time, though I have tried to cover most of the important things. And if you want more updates on this, you can periodically visit our YouTube channel, Soulmate Pharma, where we are just posting the job updates as well as updates related to pharmacovigilance courses. Means we just conduct classes or any course type where we'll just discuss different presentation. We just update videos regularly, weekly basis, so you can visit there as well. And this is the link of our channel. I have again pasted it for your sake. And anyways guys, if you have any doubt or any questions for us, please go forward, we'll take it up. I and Shobhati will try to solve the questions. So Shobhati, you can stop sharing.